Welcome. For this session, we will be discussing about the early development of the frog embryo. So let's start. In the first part of your exercise, you will be asked to examine a prepared slide of the early cleavage of the frog egg. Okay, so cleavage, this is one of the first stages of embryonic development after fertilization. So once the zygote has been formed, okay, so this is the initial process that creates your cellular building blocks to form the new organism by producing many smaller cells from the one zygote that has been formed. Okay, so this is going to be rapid mitotic division, which is why there's not enough time for for accumulating new materials for like cytoplasm, which is why each division the cells become smaller and smaller. So ultimately, as a result of cleavage, these smaller cells, okay, will differ in their cytoplasmic determinants. So cleavage division differs from other mitotic division in that there's no gap, okay? So no growth phase between divisions. So no new cytoplasm is added. But of course, your genetic material should duplicate. Otherwise, you will end up with uh, less than complete genetic material in the succeeding cells. So as the cell increases in number, they become smaller and smaller. And the embryo divides into smaller blastomeres. So called your micromeres, okay, and at the animal pole, and the larger blastomeres, which are called your macromeres, which represents now your vegetal pole, okay. So this pattern of cleavage is termed as unequal holoblastic division. So you would notice, well, it's not so obvious in this diagram, but the area, okay, that is the animal pole where the organism will develop, okay, would usually end up with smaller blastomeres or smaller cells. That's why we call it micromeres. Whereas the area, okay, which is the source of nutrition for the developing embryo, it would contain more yolk, which is why the division is slower, okay, in the vegetal pole, and they end up with larger, okay, cells. So they are now called your macromeres. Okay. In this slide, we have an overview of the diagram of the different stages, okay, during cleavage of the frog egg. So you would notice that one side is called the animal pole, and then the other side is the vegetal pole. So it is colored a bit more orangey to represent the yolk present in that area. Now, in between them, you have, okay, this is sometimes not that easy to see under the microscope, but we have a grayish area, okay, in between the animal pole and the vegetal pole. This is called your gray crescent. The gray crescent is actually opposite the point of penetration of the sperm into the egg cell. So wherever is the point of penetration, the opposite side of that, you have the gray crescent. And that will now demarcate, okay, where the first division or cleavage will happen. The first cleavage, okay, which usually happens in the frog at around 3.5 hours after fertilization, is perpendicular, okay, to the gray crescent. So as you can see here. So the first cleavage is perpendicular to the gray crescent. And then the next cleavage, okay, will happen, okay, still will happen perpendicular to the first, okay, perpendicular to the first division or cleavage, okay, but still, okay, perpendicular to the gray crescent. So as you can see, okay, the division is vertical, okay, in reference to the gray crescent. Now, the next division, the 8-cell division, would now be parallel to the gray crescent, okay? And because of this division, you end up with 8 cells, and you would notice that the cleavage is already unequal, okay? So, the cells, okay, above, okay, or superior to the third cleavage is smaller compared to the cells below it. Okay, so you now more or less have the start of your differentiation of your micromeres from your macromeres. Then the division will continue to go on, okay, until, okay, you now have a ball of cell. So here you have a blastula, okay, which is also known as a morula. Okay, because morula, that term came from the mulberry um, fruit, which is a berry fruit. Okay, so it looks like a mulberry. That's why they call it a morula. So you would notice that on the upper portion of the morula, the cells are, or the blastomeres are smaller, and then on the bottom of the morula, the blastomeres are larger. Okay? Now, these last two diagram will preempt our discussion, but this will just show you, okay, that later on, once there is now gastrulation, which is the phase after formation of the blastula, okay, there will now be, okay, an not imploding, but the macromeres, okay, the part of the yolk will start to encroach into the inner portion of the blastula, and that will now start creating a lip, okay, and later we will explain why this happens. And then at the late stage of gastrula, wherein there is now complete, okay, inversion of the, or intrusion, invagination of your macromeres, okay, then you will end up with, okay, the yolk plug, okay, which is surrounded by a lip. So the outer portion, okay, are micromeres, and your macromeres are now, okay, situated within, okay, the inner lining of your gastrula. Okay? Now, the difference between a morula and a blastula is the presence of a cavity called the blastocele. Now, the blastocele contains fluid secreted by the blastomeres. Okay? So, you would notice again in the animal pole, the blastomeres are smaller. In the vegetal pole, the blastomeres are larger. Now here, okay, we have color-coded the different cells or blastomeres of the blastomere, okay, somehow to indicate the fate or the future of each portion of this blastomere. So here in the area of the animal pole, where you have a lot of the micromeres, okay, in the future, this will form the ectoderm when we start discussing about the 3 dermal layers of the germ layer of the developing embryo. So this will later on become part of the ectoderm, which brings about, forms into your epidermis, your skin, and part of your nervous system. Now, the rest of the vegetal pole, the macromeres, okay, most of the macromeres will later end up becoming part of the endoderm, which is the innermost lining of the gut, okay? And it will also give rise to your liver and to your lungs. So that's the endoderm. So later you would find out how come it's outside right now, but later it will be form the innermost lining. Okay, that's because of the migration or movement of the cell. And then we have here two areas, okay, somewhere, okay, between the ectoderm and the endoderm. So this set of blastomeres, okay, will later become your mesoderm. Okay, so this will form the middle layer of your animal later on. And this will give rise to your muscles, bones, kidneys, blood, gonads, and others. Okay, so take note of how they align with the gray crescent. Okay, so this is the gray crescent. Now here we will require more of your 3D, okay, three-dimensional imagination. Okay, think of yourself as those CIA agents wherein in their mind, okay, they can see things moving around, okay, because we need to imagine how these cells are now going to move from one place to another. So, as we approach the stage of gastrulation, okay, so you have your blastocele, okay, which contains fluid secreted by your blastomeres, right? So, because the rate of division in your micromere is much faster than the rate of division of your macromeres, okay, it's lower here because of the presence of the yolk, okay. What happens is that the micromeres will now start to encroach on the area of the macromeres so that they will now start to envelope, okay, on both sides of the vegetal pole. Now, as you can see here, as they start to encroach on the area of the vegetal pole, so your macromeres are now, okay, being pushed into the inner portion of your gastrula. And part of this, you have the formation of a lip, okay? So the first lip that forms is called your dorsal lip, because that will form part of the dorsal layer of your embryo. So this dorsal lip, okay, will, okay, form its own cavity. So the cavity formed by the dorsal lip is called the archenteron, okay, also known as the primitive gut later on. Remember, the archenteron is a different banana from your blastocele. The blastocele was there even before gastrulation. The archenteron formed when the dorsal lip, okay, opened up during the involution of your embryo. Okay? So, the first to form would be your dorsal lip, and then the ventral lip will form later on. Now, as the archenteron starts to enlarge, okay, the blastocele will start to shrink. That is why at the late stage of your gastrulation, you barely have any blastocele left. What you have here is the archenteron. Now, when you see a cross-section of this slide, how would you know if you are looking at an archenteron or a blastocele? You look for the yolk plug. If you see a yolk plug and there's a cavity, okay, then that cavity is most probably your arc enteron. But if you don't have a yolk plug and you can see a clean circular, okay, slide or ball of cells, then that is probably your blastocele. You get it? So in the end of your gastrulation, you have the formation of your yolk plug, and then you would now be able to see that your micromeres, okay, your small blastomeres, are now located outside your developing embryo, and that becomes your ectoderm. And then as we have mentioned, the large blastomeres or the macromeres are pushed inside the innermost portion of your developing embryo, and it ends up becoming your endoderm. And then recall the small groups of cells, okay, blastomeres. They start to spread out, okay, together with the spread of your arc enteron, okay, and then now they form your mesoderm, okay. So now at the end of gastrulation, you have the formation of the 3-germ layer plus the yolk plug, okay. Okay, let's repeat that again in another view. So here on top, you have the animal pole with the micromeres. Here below, you have the vegetal pole with the macromeres. And in between them, along the gray crescent, okay, you have the future, okay, mesoderm. Okay. Now, at the start of gastrulation, okay, you have the formation of the dorsal lip. And then because of the rapid division or mitotic division of their animal pole, they start to encroach on the territory of your macromeres. So eventually, your macromeres will be pushed into the inner portion of your developing developing embryo. And as your dorsal lip starts to mature, it will open up, okay, your arc enteron. And as the arc enteron starts to become bigger, it will now, okay, cause the shrinkage of your blastocele. So there is now shrinkage of the blastocele, okay, until it disappears. And with the movement of the cell, okay, the macromeres will pull, okay, along the blastomeres that are your future mesoderm. So at the end, you have so initially you have dorsal lip, later you will develop your ventral lip, so that in the end, you will have here your yolk plug. This will not disappear. And then in the dorsal lip, that's where your arc enteron is connected. Okay, your ventral lip is a dead end sac. Then you have your mesoderm in the middle, and then your ectoderm in the outer layer. Okay. After gastrulation, okay, the first organ system to form would be the nervous system. That's why we call the next step as neurulation or the formation of the neurula. So here we have what we call the fate map. Okay, we are already familiar with the 3-germ layer, which are your endoderm, mesoderm, and ectoderm. Now, as I've mentioned, okay, in the animal pole, that's where your ectoderm is located. Okay, so later on, that will now become your epidermis and your part of your nervous system. Now, your endoderm, okay, would now become part of your egg yolk. Okay, part of your egg, sorry, will become partly your egg yolk and partly your primitive gut. Okay, your primitive gut, which will give rise to your intestines, to your liver, lungs, okay, your gut. And then here, the mesoderm, okay, you will now form your part of your kidneys, okay, muscle, your blood, heart. Okay, so that's where your that is going to be the future of your intermediate zone. Okay. Now we did keep mentioning about the gray crescent because it is closely related to your blastopore, which is now indication of where the dorsal side of your developing embryo is located. So the dorsal side of the developing embryo here in your intermediate zone, this is where your notochord will form. So the opposite side here, that is your ventral side. Okay. So here we have a more specific discussion of each region. So for the animal region, this will now become your prospective central nervous system, which includes your brain, spinal cord, sense organs, okay, and then your prospective ectoderm, which includes your epidermis of the skin. And then next, the intermediate zone, okay, that will now be your presumptive mesodermal region. So on the dorsal side, that's where your notochord will form. And then part of your foregut, okay, so foregut would be the upper part of your GI tract. And then somites, okay, which will form part of your muscles, okay, and then mesodermal lining of the cavity, okay, so the cover of your body cavity, and your kidneys and reproductive organs. And then for the vegetal pole, okay, that will now become your presumptive endoderm, which give rise to your midgut and hindgut. Okay, so that includes now your intestine up to the anus. It would be good to see the film that I have uploaded prior to going through this detailed discussion, okay, especially in neurulation, so that you would already have an idea of the movement of the cell, okay? So imagine that the cell is still circular, spherical, okay, flat. Now, on the animal side, we're talking about the animal side, okay? Now, three things will happen during neurulation. There will now be the formation of a neural tube. There will be migration of the neural crest. And then there will now be the formation of your epidermis. Okay? Now, what happens is that the cells, okay, so if this is the notochord, okay, so knowing that this is the notochord, this is the dorsal side, okay, remember the ball, okay, so this is the dorsal side, and then what happens is that the cells, okay, on both sides of your ectoderm, okay, will start to move towards the middle. Okay? So, as it moves towards the middle, it now creates a fold. Okay? So, this fold, okay, will now eventually close. So that, okay, your ectoderm, okay, on both sides will fuse together. Okay? And then here, this bright green area here, okay, we use that to demarcate the location of your neural crest. Okay? So once the ectoderm, okay, meet in the middle, okay, the neural crest will now be beneath the ectoderm. So now the ectoderm is a complete epidermis. Okay? And then below that, you have the neural crest. Okay? Which will now become part of your peripheral nervous system. And then, under that, okay, below that, you have the formation of the neural tube. Okay? Which will now form your spinal cord later on. Okay? The notochord is not part of your central nervous system. Okay? This will actually become part of your vertebral column, part of your skeletal system later on. Okay? Students always make a mistake that notochord is equal to spinal cord. Wrong. Okay? So the notochord will become the body of the vertebral column, whereas the neural tube will be the one, okay, forming the different segments of your central nervous system. Okay? And then here, okay, sandwiched between your ectoderm and neural tube, you have your neural crest. Okay? Don't mind these two things on the side. Okay? Later we will discuss more of that. Later on, the neural crest will now start to migrate. Okay? So it will detach from the ectoderm. It will now start to migrate. And once it migrates, later, it will form the spinal ganglia, which will now become part of your peripheral nervous system. Okay, let's go through neurulation again, step by step. Okay, so the neural plate, okay, extends from the dorsal lip of the blastopore to the anterior end. Okay, so here, okay, you have the ball, right? Okay, and then you now have, this is the dorsal portion of the ball, and you now have there the formation of the neural plate. Okay, so you can see that on each side of the neural plate, okay, there are now cells starting to bulge because your ectoderm is now starting to migrate towards the midline. And with it, okay, it carries um, the neural crest. Okay? So, initially, when it is still flat, wide, with only, okay, some ridges on each side, but not that still far apart from each other, we call this stage the neural plate. Now, once the ridges are close enough to each other, but it has not yet closed the neural tube, okay? So that you now have here a depression, a groove, okay? So we now call this the neural groove stage, okay? Or neural fold stage, okay? So here in the diagram, okay, so you have the neural plate, then you have the ectoderm moving towards the midline from both sides, and it pinches on the neural plate so that the neural plate will now become a groove. Okay? So with the neural fold, okay, it is continuous anteriorly to form the transverse neural fold. So, and encloses the neural groove. Okay? So, it's the increased elevation. So, there's increased elevation on both sides of the groove and bends towards one another until their edge meet and fuse, forming the neural tube. Now, once the ectoderm has fused, okay, you now have the formation of the neural tube. Usually, the closure of the neural tube will start, okay, from behind. Usually from behind. It doesn't close, okay, all at the same time. It starts off, the first one to fuse would be a bit, okay, from behind. And then the fusion will spread, okay, anteriorly and posteriorly. So anteriorly, you have an open, okay, you have an opening anteriorly. We call that opening your anterior neuropore, okay? And then posteriorly, okay, you now have the uh, neurosil, okay, that communicates with the arc enteron. So remember, your arc enteron is your primitive gut, okay? So this is now what we call the one in posterior, okay, side is the neuroenteric canal because it connects your arc enteron and neurosil. So later on, this will disappear, okay? So once you have the formation of the neural tube, okay, the neural tube will now start to separate from the overlying epidermis. And as it separates, okay, it will also loosen the neural crest so that the neural crest will now start migrating, okay, so as we have mentioned before. So later on, the neural crest, okay, will differentiate into your ganglia, okay, both of the cranial nerves and the spinal nerves. And then it will also give rise to your melanopores or chromatophores, part of the adrenal medulla, and part of the visceral skeleton. Okay? So, as for the anterior portion of the neural tube, okay, it will differentiate into the brain. Okay? And the posterior part of the neural tube will now develop into your spinal cord. So once you now have the complete neural tube formation, you now have what we call the neurula. Now, another event is occurring during neurulation. Okay? This is what we call notogenesis. So, notogenesis is the formation of your notochord, which is a completely separate entity from your neurulation because your notochord is not part of your nervous system. Okay? So, where does the notochord come from? So, the presumptive notochord material actually arises from the roof of the arc enteron. So, do you remember where your arc enteron came from? Remember your dorsal lip forming a cavity? Okay, that is your arc enteron. Now, the roof of your arc enteron, which is basically the dorsal side of your arc enteron, okay, will give rise to your notochord. Okay? So, as the epidermis is migrating outside, okay, that will also cause, okay, some migration of cells inside the developing embryo. Okay? So, the just a nice to know is the presence of the hypochordal or the subnotochordal rod, which is only present in frogs, okay, but or amphibians, and it lies, okay, on the lower portion, on the underlying endoderm, okay, of your notochord cells, but it will disappear anyway, okay, so it will immediately degenerate, so that is just nice to know. So you can see as the neural plate, okay, starts to invaginate to become your neural tube, okay, the roof of the arc enteron, okay, will now start to also pinch off, okay. So once the roof of the arc enteron separates from the lower portion, okay, the upper portion will now become your notochord, while the lower portion of your arc enteron will give rise to your gut or primitive gut. Okay? On the side, you would notice, okay, in the pink cells here, okay, these are your developing mesoderm. So they will also separate, okay, from your archenteron, okay, and then they will now become your somites, which are the fate of your mesoderm. Now, you would notice that this somite, okay, will actually expand, and then it will actually form its own cavity, okay? So, this cavity, okay, becomes your coelom, which later on becomes part of your body cavity, your abdominal cavity. So this is your abdominal cavity, okay? It arises from the separation of the two layers of your mesoderm. So your mesoderm will form a layer, and then this layer will split up into two. And in the middle, they form a cavity, and that will now become part of your coelomic cavity. Okay? So, as I've just mentioned, okay, the mesoderm, okay, will now spread, okay, in between your ectoderm and your endoderm, or your primitive gut, okay, and there are actually several segments, okay. So the part of the mesoderm closest to the neural tube, we call that your paraxial, okay, mesoderm, which are your somites. And then lateral to that, okay, you have your intermediate mesoderm. We call that intermediate because there's another lateral portion. Okay, more lateral to that would be the lateral plate mesoderm. Okay, so for the paraxial mesoderm, okay, this will form part of your dermatome, myotome, and sclerotome. So this is still further split into three. Okay, so the dermatome gives rise to your dermis. Because remember, your ectoderm only gives rise to your epidermis. So the dermis comes from the dermatome, okay, which is part of your mesoderm. Then, middle, you have your myotome, which gives rise to your striated muscle, your voluntary muscle. And then, the lateral part of this paraxial mesoderm is your sclerotome, which will now form part of your axial skeleton. Okay? So, your thorax, your vertebra, okay, your pelvis. And then, in the intermediate mesoderm, okay, later this will give rise to your kidneys and gonads. And then the most lateral, okay, of the mesoderm, this is the part of the mesoderm that will split into two layers. So this is your lateral plate mesoderm. Okay? So, the two layers formed by the lateral plate mesoderm are called your splanchnic mesoderm and somatic mesoderm. Okay? So there. Now, let's have more discussion on how the coelom is formed. So, as I've mentioned, the mesoderm layer, particularly the lateral plate mesoderm, okay, will split, okay, into your outer layer, which is the somatic layer, and the inner layer, which is called the splanchnic layer. Okay? And in between these two layers, you form a cavity, your splanchnocoel. Okay? So this will now extend downward, okay, and continue on down below the gut. Okay? So this will practically cover, okay, most of your body cavity. So the outer somatic layer, okay, combines with the ectoderm. Okay, so the combination of your somatic layer and the ectoderm, okay, that gives rise to your somatopleure, okay, or the body cavity wall, okay, somatopleure. And this gives rise to your amnion, chorion, and part of the body wall. Whereas, okay, the combination of the inner visceral layer together with the primitive gut, that will give rise to your splanchnoplure, okay, the wall of your viscera. So that will now give rise to the wall of your gut, lungs, and yolk sac. Okay? So to end the early development of the frog, okay, we just have here a frog embryo that has completed neurulation. We will have more of this when we discuss the 4 millimeter and 10 millimeter frog. But at the end of neurulation, so what are the structures that we can see? Okay, so this is a cross section. Okay, at and here we have the neural tube. Okay, and then in the middle of the neural tube, you have your neurocele, which is the cavity. Okay, and then below that, okay, ventral to that, you have the notochord. So the notochord is separate. Okay, we have here a subnotochordal rod, okay, which, as I've mentioned, only present in amphibians and will later disappear anyway. Now, in the outer layer, you have the epidermis covering the entire embryo. Okay, and then on the side of your notochord and neural tube, you have there the paraxial mesoderm. So the paraxial mesoderm, okay, will give rise to your dermatome, sclerotome, and your myotome. And then here, okay, here, this one's more obvious, this one, okay. So we have here your intermediate, okay, mesoderm, which will later give rise to your gonads and your urinary system, your kidneys, okay. And then here, okay, the most lateral, in older books, they call it a hypomere, okay. So the lateral plate, mesoderm, okay, archaically called as hypomere, okay. So at this point, it has not split up yet, okay, but you can see some lightening already in the middle. So once this splits up into two, the outer layer will become your somatic layer. The inner layer will become your splanchnic layer. And the cavity formed in the middle will become your splanchnoplure. Okay? And here, you have what remains of the archenteron. Okay? So, this is now your primitive gut. Because you remember, the notochord was pinched off from the roof of the archenteron, and then your mesoderm was pinched off from the side of your archenteron, and whatever is left, okay, becomes your primitive gut. Okay, and your yolk, okay, develops, okay, here, your yolk will remain here, and we have here another cavity formation, which will later develop to become your liver. Okay, so that's it for the early development of the frog embryo. Thank you.